Well, I've been researching it for almost 20 years and I'm still not sure I know enough to get up in front of people and talk about it, but I will. I didn't know Burney Chapman. I started researching the problem in 2000, so the year after he died. But my mentor was Chris Pollitt, and Chris always spoke very fondly of Burney Chapman. So it's titled Understanding the Basic Mechanisms That Lead to Laminitis, and I guess I'm not proclaiming that we do understand them completely, but there's definitely been a lot of progress in the last decade in particular, and I think we do know more now, and we're actually starting to be able to put that evidence into our efforts at actually preventing and treating this problem. A little bit of an anatomy refresher. There are 600 odd primary epidermal lamellae, so like leaves of a book that interdigitate between the dermal side, the bone side, and the epidermal side, which is the hoof side, spread right round into the bars. My PhD student back in Australia, Simon Stokes, proudly proclaimed to me that the volume of that tissue is about 45 mils. So if you take 1 foot's lamellae and stuff it into a container, it's about exactly the same volume as your average shot glass of whiskey. And serendipitously, a pony shot, I understand, is 30 mils. So 1 ounce, so a little bit less. It's not enough just to have those primaries. They're divided up to 200 times into secondary epidermal lamellae, which are these smaller projections, and they increase the surface area of attachment massively. So there's a meter squared of surface area of attachment in each foot. And without them, it's 0.006, actually, sorry, 0.006 meters squared. And cows actually don't have secondaries, they only have these primaries. And so the cow foot is a lot different. They rely much more on the digital cushion for bearing. There's 2 claws, the mechanics are a lot different. Arguably, also a little bit less athletic, the cow. These cells that line those secondary epidermal lamellae are very specialized, and we call them epidermal basal cells. Their only job is to hang on. They don't have a direct blood supply, they're relying on blood diffusing from the nearby dermis to deliver glucose mostly. And if we really drill down on these cells, what's holding this whole tissue together is these sort of spot welds which attach these cells to the basement membrane. And these other welds that attach these cells to each other, these are called desmosomes, and these are called hemidesmosomes, but they're essentially welds. And although these cells don't, muscle cells contract and shorten up and use energy to do that, and these cells don't do that. These cells just sit there holding on. But they're anything but static in terms of their metabolism. They consume a lot of energy. It's an energy requiring task for them just to maintain these attachments. And we did some experiments years ago, Chris Pollitt and I, and Uwe Wattel, looking at the consumption of glucose. And if you measure the blood coming out of the head and compare it to the blood coming out of the foot takes more glucose per unit blood out than the brain does, which gives you an idea about what's more important to the horse, I guess, the foot or the brain. So nourishing that tissue is important, and there's a blood supply that has some redundancy to it. Both sides are anastomosed down here, the terminal arch, and there's also anastomoses at the circumflex arteries of the sole and the coronary band. The arteries perforate that sort of Swiss cheese nature of the distal phalanx, and that's what supplies a lot of the lamellar tissue is these arteries that are perforating through the bone via the terminal arch. The venous side is even more extensive, and there's a lot more joins in the venous side and a lot more sinuses and a lot more areas where blood can collect. And that's particularly true up around the collateral cartilages, the ungular cartilages. And there aren't really, normally in a limb or another part of the body, there are a lot of valves in the veins, which means the blood can only go one direction, back towards the heart. That's not really so in the foot. Within the capsule itself, there aren't really many valves, so there's some reliance on the action of weight bearing and unweight bearing and that sort of pumping action to push blood back out of the foot and back up the leg. And there aren't just a lot of connections between veins and other veins. There's also connections between the arterial side coming from the heart and the venous side that are direct. And these are these arteriovenous anastomoses, big words for just joins between the arterial side and the venous side. These are typical of parts of the body that are involved in temperature regulation. So you find these arteriovenous anastomoses, or AVAs, you find them in elephant ears, in rabbit ears, in our fingers, in our fingertips, and you find them in horse feet. And what they allow us to do is to turn blood flow on really quickly. It bypasses the smaller blood flow beds, so it bypasses the tissue, doesn't deliver nutrients. What it does do is deliver a lot of blood, generally to divert warm blood to that part of the body quickly. And all this is sandwiched between a rock and a hard place, or two hard places, I guess, the bone and the hoof wall. So it's set up in some ways for to be a little bit precarious. Laminitis, in essence, is just failure of that attachment, failure of that normal suspensory function that the lamellae serve to hold the bone up inside the hoof capsule. And consequences of that are that since the bone is no longer suspended, we start to get bearing on the distal surfaces of the distal phalanx, which are not designed to do that. And we also get disruption of the normal growth zones, the coronary and terminal papillae, which distorts and disturbs hoof growth from then on. That separation happens between, this is normal here, this is separation between the epidermal side and the dermal side, which tends to happen at that junction and classically happens at this basement membrane, which is this purple line, the division between the two. And this basement membrane starts to slip off like the fingers of a glove. Every now and again when we're looking at research samples, we're reminded of the importance of the pull of mechanical distraction. When you look at a lot of tissues, you sometimes see these islands of epidermal cells out in the dermis, and these are left over from a previous injury, more than likely. And even though this horse has got some acute laminitis and a lot of destruction here, these islands of epidermal cells that aren't connected to anything, they're not part of the weight-bearing apparatus, are sitting here sort of quite happily, like sort of like an island that's missed the hurricane that's come past through. So there's a lot of importance, there's a lot of contribution in this lesion of mechanical distraction, of force, which is unique to our tissue. Normally the lamellae don't do a lot of dividing, they don't contribute to growth of the hoof really, but if you create an injury, if you do a wall strip, this was a paper Dr. Pollett did in 2004, where he did some wall strips. When you do a wall strip, rather than breaking the connection between the epidermal and the dermal side, you tend to break the tips of the primaries off. And what you see over there, this is 2 days later, you see rapid repopulation of that basement membrane, which is brown in these pictures, until 3 and 4 days later, it's almost repopulated back to its normal architecture again. So these this tissue retains an enormous capability to divide and heal. The problem in laminitis is that, we'll see later, is that it can't get back to that normal architecture again. The progression in laminitis depends a lot on the initial severity of the problem. So if it's severe and widespread and involves most of the foot, then we tend to get widespread failure of that attachment, and that's your classic sinker type foot. If it's a little bit less severe, then just like the wall strip, we get repopulation of that basement membrane, but the basement membrane is now damaged. And rather than going back to that lovely feather-like architecture, we get this disrupted, kind of ugly mess that is not only in 2 dimensions, but 3 dimensions. A lot of these secondary epidermal lamellae aren't connected anymore. They're just islands, like those ones we saw before, so they're not contributing to the mechanical support of the bone anymore. And largely those changes are irreversible. And I say that with a bit of an asterisk because I've got another lecture tomorrow afternoon, and we'll address that point again, whether they are irreversible or not. But classically, we feel like they're irreversible. Once we get to this architecture, we can never go back to that because we lose that fine anatomy of the secondaries, and we're sitting there. And this tissue, no matter how it's damaged, this is natural laminitis. This is kind of the hoof side. This is a whole bunch of cap horn forming here, contributing to the lamellar wedge. This is experimental laminitis, 7 days later, very similar appearance to that. If you do a biopsy, which we sometimes do, in a normal foot, and come back and look at that tissue a year later, it looks the same. It's sort of like a one-trick pony. No matter how you damage it, what does it do? It proliferates, it fills the void, it creates a lot of this cap horn, which is kind of like scar epidermal tissue, and that's what contributes a lot to the lamellar wedge. It's not only just the descending bone, the rotating bone, it's also that this tissue becomes proliferative and proud. So when I first started in 2000, we thought all laminitis was essentially the same, no matter sort of what the cause, but now we recognise that there are 3 major forms, and these forms are probably have different mechanisms to each other. There's the sick horses, there's the fat horses, and there's the ones that are lame. So this is sepsis-induced laminitis, so sick horses with disease, insulin-mediated or endocrinopathic, which is a big, it's kind of a stupid word, I think. I like to call it insulin-mediated, and supporting limb laminitis, the sore foot, sore opposite limb, too much weight on one leg. And although we used to think it was kind of all the same, we realised in particular that these forms are actually quite different. This classic sort of sepsis-associated, acute laminitis in the sick horse, where you've got a lot of that basement membrane destruction that we were just talking about. We actually don't see that as much in the insulin-mediated cases, which are the majority of cases that we see, as vets and as farriers. In those cases, it's much more lengthening and stretching of the cells themselves, and not necessarily more cells, but actually stretching of those secondary epidermal lamellae, which is interesting and an important point. So I think we're starting to get improved understanding of what events lead to each form of laminitis, and that's kind of what I'm going to talk about. I Googled the same pictures as Domenico of Barbaro. I was a resident training to be a specialist at New Bolton when Barbaro arrived in 2007, and I remember it vividly because I had some 23-year-old paint horse with choke that I was treating, and that was my the glamorous thing I was doing when Barbaro arrived in the hospital. But he had a terrible hind limb fracture, and the facilities there are just incredible. Dean Richardson was able to reconstruct that that limb, which is, these days, with the expertise and the equipment and the facilities for recovery that these horses have, the fixing of these complex fractures is quite possible. It is quite possible. That horse was there for a while, and there was a lot of well-wishers who sent cards. They sent food, which was very welcome for someone like me as a starving young resident. Someone sent a statue of St. Francis of Assisi, which had holy water in it. And there was 3 horses with laminitis up in the ICU, and I had one called the Arts Place, actually. Some of you may remember as a sire of sires on the standardbred side, who had really bad laminitis. He lost both his front capsules. And there was another horse suffering from endocrine laminitis there as well. And I sprinkled each of these 3 horses with holy water, and all 3 are dead. So I can pretty confidently say that we've ruled out holy water as one potential treatment. So Barbaro is now a statue down at Churchill Downs, and the whole reason I'm back at New Bolton Centre, I came back there this year, is because of the job that was created by Roy and Gretchen Jackson in memory of Barbaro. So my position as a researcher at New Bolton Centre is funded by their donation in his name, which is serendipitous, I think. We don't know a lot about supporting limb laminitis. There's not a lot published. This is about the only study that's published, and of all these horses, 113 horses that had casts on their limbs for different reasons, 12% of them developed supporting limb laminitis, so a bit more than 1 in 10. And body weight, so the heavier ones, were more likely to get it, and the length of time they had the cast on was also important. So, that's not very surprising, but this is the first study to actually show that the heavier they were, the more likely they were to get it, and the longer their problem went on, the more likely that it was that they would get it. The problem with these is, in my hands, I think they approach 100% almost. The problem is they've got this opposite limb problem, which is still severe. They found her in the good leg. They're often a euthanasia proposition at that point. They're often rapid and severe, and they sink. This is just a mid-cannon fracture, complete fracture in a racehorse, which, again, the importance of supporting limb laminitis is we don't often attempt to treat this type of problem because we think they're probably going to founder. And when you do go to treat them, this is the surgeon back in Queensland. They always show me these pictures, and they're very proud of the job, and it's fantastic. I call it a high steel to bone ratio, which generally means they're going to founder. In my experience, in this one, 24 hours later, did, and really dramatic. We had radiographed this one only hours before, and then I walked past this horse at about 6:30 at night. I was about to go home, and saw it sweating and standing on this leg again, and we radiographed it immediately and saw that it had just sunk. So the threat of this means we don't often attempt to fix these types of fractures, but more importantly, what they're discovering at Penn, the pathologists are looking at a lot of tissues from horses that were put down because their repair failed, or because they didn't go to surgery, but they had a painful problem in an individual limb. And a lot of those horses, even though they have no signs of it, have mild laminitic lesions already starting. And I think we'd be really surprised that a lot of horses have different degrees of laminitis that aren't necessarily showing it as lameness when they have a painful condition in one limb. And it's not always just the opposite limb, it's often multiple limbs, which I think is really interesting. So what are the possible mechanisms? Could this be purely a mechanical overload problem? Could it be a blood flow problem? Could it be that They're getting a generalized, like just regular laminitis type insult that's affecting their whole body, and it's just worse on that leg because they're putting more weight on it. Or is it some sort of complex signaling thing? And this is just a complicated slide telling a story about a study in rats where they fracture the femur of these rats, the tibia, which is the shin bone of the rats, and in the weeks after that, they actually start to get growth of the epidermal tissue of the paw pad, and proliferation and overgrowth and thickening. And what that is, it's a signaling event happening that's associated with the pain, and the pain pathways in that particular limb signaling, causing signals to go via the bloodstream to that epidermal tissue and causing it to proliferate, which has interesting implications for our problem. In rats, interestingly, it's the same leg that gets the problem. But if it was on a milder scale and they're standing mostly on that leg, the only thing that goes against that is we definitely see supporting limb laminitis in horses that have non-painful problems. This horse has radial nerve paralysis, and we see it even in that problem, which is not a painful thing. As Domenico was explaining, the ground reaction forces in feet can greatly exceed their entire total body weight at faster gaits and at canter, gallop, and jumping. It's hard to believe that we could overwhelm the mechanical properties of the tissue, but this is over days or weeks, sometimes, or even months, which makes you feel that perhaps that could be a factor. It was a farrier, another ingenious farrier, Van Krayenberg, a South African farrier, who first showed in cadaver legs that there was interruption of blood supply. Actually, it was in live horses, interruption of blood supply under load, and he used fluoroscopy and arterial injections of contrast. And those were in thoroughbreds and Arabian horses. And Chris Pollitt repeated that and showed that under load, there wasn't flow into the capsular area. When you ultrasound the palmar-digital arteries, and this is the palmar-digital artery here, the black zone, and you lift the opposite leg, which we're going to do here, those arteries shut down. They shut down at the level of the pastern, sometimes completely, sometimes partially. Sometimes, remember, there's redundancy, there's both sides, which join at the bottom. Sometimes one side and not the other. So there's a lot of variation between horses. We looked to repeat that study using new technology, and when we first started, this is Chris Pollitt here with our jig that's designed to take these cadaver legs into the CT machine. When we first started doing this, we didn't have access to a CT machine in a veterinary hospital, so we used to turn up at the human hospital with a bag of dead legs and a couple of bottles of red wine under each arm, and then we got access to our own CT. And what we would do, we'd hook up the arterial side to a contrast and an automated pump, and we actually took some standardbred horses and we looked at values for fetlock joint angle in particular in their live state and in their cadaver limbs. We replicated that. We put some tension on the deep flexor using a spring balance, and we looked at what was normal perfusion. And this is an unloaded, it's not perfusion, this is vascular contrast, which is important to remember. All we're doing is pumping contrast into the vessels, but we're pumping into the arterial side, so it's different to a venogram, okay? When these horses are loaded to what the equivalent of square standing would be, we already see quite a lot of a deficit in perfusion. Even just loading to square standing is enough to interrupt blood flow, and we actually start to get cut off at the palmar areas of the palmar digital arteries at the level just above the heel branch, but also as they enter the back of the bone and the terminal and the solear canal. That gets more prominent as you increase the load, so it's equivalent to unilateral weight bearing. You can really see now the attenuation and actually the gap in contrast fill at the distal ends of the palmar-digital arteries there. And that gets more severe as you progress to maximal load, so galloping type, lead leg force. What happens when you put a heel wedge on it, which was one of the questions that I was quite interested in answering. It didn't seem to affect it much in our study. And in fact, when we looked at contrast fill, this is a score, so more is better. When they don't have any load on them, when they have normal square standing load, when they have unilateral weight bearing load, and when they have maximal load, it's kind of quite a neat graph in terms of the score, and that's the score around the dorsal aspect of the foot. And putting a wedge, a heel wedge on them at 20 odd degrees didn't really make much of a difference. And that's in a static cadaver leg, okay. So there's definitely evidence of arterial occlusion at increasing limb loads that affects contrast fill in cadaver legs, and even just at square standing force. So that starts me thinking that, you know, a horse can't just stand there statically for its entire life, which they never do. They always do a bit of this, right? And it starts to get you thinking that really do need to do some of that just to get the blood flowing and get the dorsal tissue perfusing. So what happens in the live horse and what is the variability? We've got access now to this robotic imaging center at Penn, which is really cool because you can get three-dimensional CTs of feet in a minute in a standing horse, if you can get them to stand still. So we're planning on repeating this experiment with arterial contrast in standing horses, which hopefully if I don't embarrass myself at the bar and I get invited back, we might be able to talk about next time. I'm also hoping to replace Pat Riley with a robot one day, which he's not particularly happy about. Don't tell him that. So looking at blood perfusion is probably not enough. When we think about what the importance of blood perfusion is, it's about supplying energy to the tissue. So how can we study energy balance in the live horse foot? We adapted this technique it's called tissue microdialysis. It's just a little tiny probe. It's about the same size as the smallest blood vessel. It's about the same size as a little bit larger than one of your hairs. They put these in, they've been putting them in people's brains for a while when they have traumatic brain injury to monitor the energy state of the brain to see if they need to intervene in different ways. And essentially what we look for in the tissue are just basic energy metabolites like glucose, which is brought in by the blood, lactate, which is produced by breakdown of glucose, pyruvate, which goes into the oxidative side, and the ratios of those things. And if you cut off the blood supply, this is basically a strangulation experiment here in a piglet. The glucose drops in the tissue, the lactate goes up, and this lactate to pyruvate ratio, LP ratio, goes up, and that green one is the important one. That's the marker that there's not enough blood flow, that the energy balance of the tissue is gone bad. So we adapted this to the horse foot, which, these things are designed for laboratory experiments or people sitting still in a bed. So it took a while and it was an expensive proposition to adapt to the horse foot. But we're now able to we put probes both in the sublamellar dermis and also between the lamellae and the lamellar dermis, and this is the hoof wall here. And you're able to take samples as often as every 10 minutes or even every 5 minutes and look at the balance of these things over time as you do things to the foot, which is really cool. And a way to look directly at how much blood is flowing is to put something in the side that you're perfusing in and measure how much comes out the other side. And if a lot less comes out, it means that there's more blood flow to the tissue. So this urea clearance is a measure of blood flow. So when we put a tourniquet on a leg like we would for a venogram or for a regional perfusion, the urea goes up, which means the perfusion has gone down. It's the opposite. The glucose drops down, just like the piglet strangulation experiment. The lactate goes up, and the ratios go up. If we walk the horse, urea goes down, which means the perfusion has increased, the blood flow has increased. The glucose really massively goes up in the lamellar tissue when the horse is walking. And the interesting thing is that that does not happen in the skin. So this was the skin over the tailbase that we used for comparison. So it's not just when they're walking around that their heart rate is increased and the general blood flow is increased. This is a lamella-specific event that's occurring. What happens when you just pick one leg up and put it down like that? When the horse is standing still, you get a mild change in the urea clearance, a mild increase maybe in perfusion, and a significant increase in glucose availability within the tissue, but it's much less profound than when they're actually undergoing a weight-bearing cycle, okay, which is important. We actually, one of the first things I did then was I thought, we need to see what happens when these horses are bearing weight just on one leg. And we taught some horses to accept a knee hobble for an hour, and we saw absolutely no change. But when we looked back, these horses that had been trained to this knee hobble were pretty much hopping around at the same rate as they would normally cycle their weight. So we needed a better way to model increased preferential weight bearing on one leg. And Jim Belknap at Ohio State University came up with just using a platform shoe with a point essentially. And with this shoe on, the horse can bear some weight, but they can't really comfortably put full weight on it all the time. And so over time, they preferentially weight bear on the opposite limb. And in this introductory experiment that was only 48 hours long, we already saw that there was some evidence that using this hypoxic ischemic factor, which is a marker of low blood flow, in the tissue afterwards, we started to see that there was some evidence that perhaps this weight bearing was interfering with perfusion. So we designed a microdialysis study to look at the same thing, and they wore the shoe, they stood on a forced platform, and with this forced platform over time we could look at the number of times. So this is the weight-bearing limb here, and these are instances of load cycling, of offloading here, and you can use software to count the number of times that they're offloading. We blocked these horses continuously so they felt no pain at all on the weight-bearing limb. They had a continuous infusion of local anesthetic next to the nerves. And they tolerated this remarkably well. They got bored in that we did it for 96 hours for 4 days. So there was a lot of horse entertainment involved, but it was well tolerated. These are the heart rates control versus support limb, which were really not significantly different at all. The what, sorry? I'll get to stuff like that in a second. You're very eager, I can see. How do they distribute their weight? Normal situation here. This is the support limb here, and it's probably more accurately looked at as a percentage of body weight. So these are the forelimbs here in the normal horse. So about 28% is normal in the forelegs, and 21, 22% in the hind legs. And this is our preferential weight-bearing shoe horses. So the shoe's on this foot, and this is the support limb. So there's 10% more, or 50 kilograms, or 100 pounds more weight on that support limb than a normal standing control horse. 10% body weight more, 50 kilograms. It's not a massive amount. This is average body weight, average over time. So what did we see? What did we see across the 96 hours in terms of energy balance in that tissue? And remember, I said that lactate pyruvate ratio, that the green one in the other slide, that was the important marker of energy balance in the tissue. And this is the control. This is the lamella probe. This is the control horses here, very tightly regulated, kept below about 30 for the entire time in healthy tissue. What we saw in our preferential weight-bearing feet was from about 36 hours onwards, we saw a really remarkable climb. And in brain, anything above that red line is considered ischemic, is considered negative energy balance and cell death. So we really got massive increases in the second half of the experiment. And the really interesting thing that kind of threw us off quite a lot is we had a probe sitting in the sublamellar dermis closer to the bone in the same feet for the whole time, and we did not see any of that occur in that zone. It's only between the lamellae. So it's not a whole foot phenomenon, it's just a lamella phenomenon. This is actually the same feet. So this is sublamellar dermis, and this is lamella here. And along with that, the urea clearance. This is the clearance, so it dropped less perfusion in the lamella zone compared to sublamellar. And so when we started to look at the load cycling frequency, and I thought, well, it's going to be really interesting. We know that there's more weight on the leg. It's going to be really interesting to look at whether they're cycling less frequently in that loaded leg to see if that's contributing. And this is control versus support limb, and it looked pretty unexciting on first glance for sure. The interesting thing is that in the control feet, they actually increased the frequency. And this is, probably nighttime, daytime, nighttime, across the 4 days in terms of how frequently they're shifting their weight, okay? And that increased in the normal horses over the course of the experiment as they got more bored, more than anything. And the interesting thing was in the supporting limb horses, they started out, the shoe goes on about here, they started out cycling much more frequently when the shoe first goes on. And their frequency actually drops over time. And when you look at the mean lines, the trend lines, they actually cross at about that 36-hour point, which is where we start to see problems. So it seems that it's a combination of increased weight on that leg, and they really do need to cycle more if there's more weight on it. And as they start to cycle less on it, it starts to cause problems. And we saw in individual horses that the blue line is the frequency of weight shifting. The orange line is that lactate pyruvate ratio. As they drop, they start to get an increase in that ratio, which means they're starting to become ischemic. So as they cycle less frequently, that's when they tend to get the issues in the individual horses. So this supports the theory that low blood flow plays a role in supporting limb laminitis, but on the question of inflammatory markers and other things, that tissue is being analysed at the moment, so I can't tell you yet. But there's more than meets the eye here. There are changes in the other feet. Nothing's ever as simple as you'd like it to be. But the implications this has for prevention, I'm going to talk about tomorrow. This has got nothing to do with feet, but we can't sit here and talk about feet the whole time because we'll go mad, right? I moved into this house when I moved back for this job, my driveway is here. I moved into this house, and the week I moved in, this 250-year-old white oak tree fell over, massive crack in the middle of the night, and heard a bunch of chainsaws going and wondered what was going on, and found this. It was actually over the rail line. And I'm a keen amateur woodworker. I'm not particularly good, but I try very hard. And I know there's probably quite a few of you who are interested in woodwork. So when I heard that the guys who owned this little strip of land here were dragging most of this away for firewood, I got very upset and asked them kindly if they could drop off a couple of pieces. And the guy was great. He had to counterbalance it. These were some of the biggest pieces he could pick up, and they're absolutely massive. To put it in perspective, as my wife and child, my oldest son Edward volunteered to help chop it with his saw, but then I actually decided to. I'd never done any milling of my own, but you just got to buy the biggest chainsaw you can get and get yourself an Alaskan mill, and it's very addictive, I'd have to say. Slabbing large pieces of wood and arranging your children on top of them for photograph. Awesome fun. The stuff's drying in my backyard now. There's a larger piece of log there. There's my youngest son, Max. So this will probably be, hopefully, be a kitchen island one day. But this is all practice for the big game, because there's a piece still in the hillside that's about 14 feet long, that is 58 inches in diameter, 5 foot, and I need to try and mill that. And my wife was at the local hardware store, the Ace Hardware, picking up some dog food and other things, and asked my 3-year-old son if there was anything else we needed, and he promptly answered that Daddy needs a new chainsaw. He needs an MS-880, which you got to be very proud of your son for coming up with that. Back to the boring stuff. So what about the sick ones? So we've talked a bit about what we're starting to learn about the ones that are putting too much weight on one leg. What about the horses that are sick with metritis, with diarrhea, with colitis, retained membranes, pneumonia? Endotoxemia is a risk factor for laminitis, and these horses are septic. Septic just means they're inflamed in their whole body because they've got a bacterial infection or an overgrowth in some region of their body. And people get organ damage from sepsis as well and die from it. And the organ damage, they don't, actually, funnily enough, their fingernails fall off sometimes, but they don't tend to worry as much about that as we do with horse feet. What they worry about more is failure of the lungs and failure of the kidneys and other organs. And the reasons why this happens has been a focus of human research for a long time, and they still don't know, which makes us feel, I guess, a little bit better. What are the, there's a lot of inflammation, there's programmed cell death, there's energy failure, but they don't really know. And this slide's just intentionally complicated. We studied this form of the disease for a long time, and there's a lot of things going on. Chris Pollock got excited about MMPs, and that's kind of proved not to be as important as he originally thought. Most of it, actually, though, is not as important, because we can know now that we can prevent it reliably with foot cooling, which is something that they can't do as easily in human sepsis. And as we're studying the reasons why, we're kind of almost doing it in reverse now. We're looking at experiments where we cool one foot and not the other, and looking at what's going on. We start to see that there are, in the cool feet, we're preventing a lot of these inflammatory markers from increasing. And recently, and we haven't published this yet, but we've also seen that same sort of energy failure we see later It's not an initiator, but it's a bit later in the development of this form of laminitis. We also see some of that increase in lactate-pyruvate ratio, which is prevented in the cool feet. Cooling has an effect on metabolism that spares tissue from negative energy balance. So, the jury's out on what exactly causes that form of it, but it's becoming less important because we can stop it from happening by cooling. So what about the majority of cases that we see as vets and farriers, these, the fat ponies and the old horses with Cushing's, essentially. So metabolic syndrome, and Cushing's disease horses. And these encompasses pasture-associated laminitis. The dysregulation of insulin, so the lack of insulin control, is the key in those horses as well. And insulin is the key in Cushing's disease as well. And we knew that a long time ago. This was a 2004 paper where in cases of Cushing's, which is PPID, horses with high insulin didn't survive and horses with low insulin survived. We even knew it kind of back then. Insulin is everything, but it wasn't until this model was developed, and this was again at Chris Pollitt's University of Queensland lab, 2007, you pump a lot of insulin into a pony and you can cause laminitis lesions. You pump a lot of insulin into a standardbred horse, which is classically not associated with this form of laminitis. You can also get laminitis. And people started to argue, well, 1,000, that's way too high for insulin. It doesn't mimic the situation in natural horses. But, the interesting thing is that you can produce those sorts of levels of insulin, 200 to 1,000, quite easily with high non-structural carbohydrate diets, which is what's happening in the field. So how does, what's insulin all about and how is insulin produced? Sugar goes in. This is I own this standardbred horse. I own 2 standardbred horses. I think I feel bad for standardbred horses because we use them a lot in research. So I own a couple of them around the world that are retired and are not useful for anything except burning money. But this guy's called Buster. And he eats the sugar gets broken down, some glucose gets absorbed, the blood glucose goes up. There's hormones that get released along with that. They're called incretins. They're local gut hormones that both those local gut hormones, the incretins, and the blood glucose act on the pancreas, which then produces insulin. And that insulin feeds back on the glucose to push it into the cells and lower your blood glucose. And that's kind of the loop in a nutshell. That's the normal situation. So how do we get excessive insulin production? Typically in the overweight horse or the overweight person, what happens with time is that negative feedback loop becomes less active. And this is what we call insulin resistance. This is insulin resistance. The tissues they're not as sensitive to insulin anymore. It's not driving the glucose into the cells as much. And as a consequence, the glucose goes up. That pushes the insulin up more just to push the glucose back to normal levels. And that high insulin is what acts on the feet. So this is the insulin resistance situation. But you can actually also get it without insulin resistance. And you see this in non-overweight horses and ponies, and particular breeds of ponies that are not overweight. They don't have to be overweight. They don't have to be insulin resistant. They can just have a tendency to produce a lot more of these incretins, absorb a lot more of the glucose from the diet, and produce a lot more insulin as a consequence. And that's what acts on the feet without insulin resistance. So they don't have to be fat. And there's a really neat experiment that demonstrated this out of the group in Melbourne, where they took standardbred horses and Andalusians and ponies, and I've color-coded it. And these are the feeds at twice daily intervals, and this is the blood glucose going up, which is not a lot different. The Andalusian, maybe a little bit higher, but not a great difference. What's interesting is the insulin difference. So this is the insulin, this is the standardbred here. Not much of an insulin spike at all, but look at the Andalusian and the pony. Massive insulin spikes, and this is the exact same feed, What's driving this? Incretin production to an extent. So this is one of the incretin hormones, and fairly massive increases in the Andalusian and the pony compared to the standardbred. So some horses and some ponies are just innately programmed to produce a lot more insulin, and it's kind of inside, and it's part of their makeup. But there are no insulin receptors in our tissue, so what causes laminitis? Insulin can activate a different receptor called the insulin-like growth factor receptor, which there are a lot of in the foot. And now there's fairly strong evidence that it's activation of that insulin-like growth factor receptor that causes the cells to want to divide and break down their adhesions and stretch and rearrange their skeleton so that they're not as stiff anymore. And that stretch and proliferation is what characterizes this form of laminitis. It's lengthening, stretch, and not necessarily that basement membrane breakdown and that type of thing initially. Really interestingly, this growth factor signaling is a common event in all three forms of laminitis, and we talk about how there's different pathways. My good friend and collaborator at Ohio State University, this is his data, Professor James Belknap. This is happening in all three forms of the disease, so there's potentially at the final event some commonality between all three forms, and interestingly, potentially this also could be therapeutically blocked. So I'll leave the rest for tomorrow. I hope you guys can see me again tomorrow when we talk about prevention and treatment. This is Pat Riley and I. I'm pretending to use the Dremel. This is when we had more brown hair about 10 years ago. Thank you very much, and invite any questions that you have.
Can you hear me? Yeah. First of all, great talk, Doctor. I was wondering with the support limb, I have to say that I would let horses sometimes go 7 to 14 days before I started getting worried about supporting limb. Obviously, that's way late. So what can a farrier do on these horses when they just have a simple lameness, an abscess or something like that, where the horse is not putting a whole lot of weight on them? What can they do to help with these horses?
Dr. Riley, we'll talk about that tomorrow. I got to have something up the sleeve.
Thanks for the talk. You said you blocked the feet when you were looking for supporting limb laminitis. Does the blocking not influence the pain, and the pain is the cascade that also activates the inflammation? How much does that influence it?
The blocking takes away all the pain signaling out of that foot. So that's the weight-bearing foot. Mm-hmm. So, for us that was important from a welfare standpoint. We didn't want them to be uncomfortable. But I think it's also important from the model standpoint because, what stimulates a normal horse to actually cycle on and off their limbs is probably discomfort in that leg. And you guys as farriers and shoers and trimmers would understand that if you lift a limb, you have, in most standardbred horses, you've got about 8 minutes before they want to put their foot down. And if they hop, they're usually okay for another few minutes, There's some discomfort that stimulates the need and the want to cycle. And I think what happens in a lot of natural cases of supporting limb laminitis is what's going on in the other leg combined with a lot of the medications that they're on may take away or overwhelm the stimulus for them to actually cycle off that uncomfortable support limb, and that's what we have to combat. But if your question is more directed to will that modify the signaling,
Yes.
we've done it in our control horses too, so we've got that controlled for when we go to look at the inflammatory markers. The normal horses that were standing square also had that one limb blocked continuously for the entire time. So we can look at whether there's any difference, but I don't have those results yet.
But don't you think that the inflammation is going to be enhanced when they do feel the pain?
But if they're blocked, oh, you mean, will it be worse from, I don't know. You know, I don't think we have evidence for that in horses, that inflammation comes from pain directly from signaling. It could be. There's definitely a stress response and a lot of things that are, you know, when we were trying to develop this model, you know, welfare was the number one issue for us, so it couldn't be a painful model, it just had to be a non-painful model. But yeah, there are a lot of other signaling things going on in the natural case, like pain, that probably contribute to things that we can't produce in a model. Yeah.
Just a diagnostic question that I've run into all the time. Whenever I'm testing for insulin resistance, and I send my results to Cornell, as many do, and it's extremely rare to have an elevated blood glucose in a horse, period. It almost never, and when it does, you're in deep doo-doo.
Yeah.
Can you explain that?
Why is that so? It's the overwhelming ability for the horse pancreas to produce enough insulin, full stop. So, you know, this is not like type 2 diabetes in people. So type 2 diabetes in people, often you get insulin resistant, and you start to produce more insulin, and you're still eating donuts, and your sugars go up in your blood. But it gets to a point where your pancreas cannot produce enough insulin anymore to counter that absorbed glucose. The horse never really ever gets to that point. They have a super pancreas. They just keep making more and more and more insulin. And it's so very rare to see an actual type 2 diabetic horse or pony because of that factor. So they always produce enough insulin to keep their blood glucose under control and to keep it down.
You know, we've had a few people out in the literature that are saying that they're monitoring it with a glucometer, so that, and I was just looking at.
Yeah,
then they're all healthy that way.
Yeah, the blood glucose is it's not very useful. I agree completely. It's all about insulin. Yeah.
Thank you.
Thanks.
Wonderful lecture.
Thank you.
Any other questions?
We have one over here.
So in horses that you expect to be septic, post-op colics and that sort of thing, are you doing cooling of the feet prophylactically?
Yeah.
When do you start that generally?
Straight away.
Right away. That's amazing.
So in colitis horses and horses that appear endotoxemic or have a low white cell count on blood, we start cooling them immediately.
When you had the probes in the lamella and inside there, and you didn't see a difference in the blood flow closer to the bone, correct?
We do. I didn't really have enough time to go into. We do in the normal foot to an extent. There's a bit less as you get out, as you'd expect, between the lamellae towards the hoof wall. There's less blood. There's more glucose being taken up by the cells. But in that experiment, no, we didn't see a change in that probe that was positioned next to the bone.
So that was dorsal, correct?
Yes.
Would you expect any difference peripherally around the foot or also in the sole corium?
Yeah, I don't know the answer to that. And the questions that I would also like to answer. It's logistically very difficult to just put the two probes in and do the experiment. So at the moment we're limited to that, but and we're limited to the dorsal wall. But I would like to see the other parts of the foot. The way I look at it though, the sublamellar probe, the one that's next to the bone, is more representative of the whole foot because you've got a lot of large vessel anastomoses, and some of the subsequent experiments we've done have probably shown us that what's happening in that probe site is probably more indicative of what's happening in the sole corium, because you get a lot of the venous drainage come up in that sublamellar zone. So I think all it's telling us is that it's not a whole foot phenomenon, this lack of blood flow.
Right.
And why that's the case, you know, I think sometimes it's convenient to look at it and try and oversimplify it, but I think there's more factors that influence flow through the lamellae than you initially think. It's not just the amount of arterial blood that's coming down, it's also how easy it is for the venous side to drain out. And blood will take the path of least resistance through the foot. So if the post-capillary pressure is high because the veins aren't draining back up, you know, I think that could also contribute to lack of flow through those lamellar vessels. And when you're watching it in real time, it's a rapid, it just switches off. It's like those vessels collapse and they never open again. You know, and I think it's a tipping of the balance of pressure that occurs. That's probably due to a lot of different factors.
Okay. Thank you.
Hey, let's give him a hand. Thank you, Dr. Van Epp.
Thank you.